All right. Happy Friday. So the next line of speakers we have Dr. Isabel Deganier and Dr. Yezhang Ding. And today's speaker is Dr. Erich Grotewold. It's my great pleasure to introduce Dr. Erich Grotewold. He's a distinguished Foundation Research Professor in the Department of Biochemistry and Molecular Biology at Michigan State University. Dr. Grotewold completed his PhD in chemistry at the University of Buenos Aires in Argentina, followed by a postdoctoral training at Cold Spring Harbor Laboratory. And he began his independent career as an assistant professor at the Ohio State University, where he rose to full professor. And during his tenure at Ohio State, he also provided significant leadership as director of the Arabidopsis Biological Resource Center and director of the Center for Applied Plant Sciences. In 2017, Dr. Grotewold joined Michigan State University, where he later served as a chair for the Department of Biochemistry and Molecular Biology. And Dr. Grotewold is internationally recognized for his pioneering contributions to advance our understanding in phenylpropanoid biosynthesis, especially flavonoid compounds and floral pigment genetics, and also plant gene regulatory networks. And his leadership has been honored through multiple awards, including election as a fellow of the American Association for the Advancement of Sciences, and also as a fellow of the American Society of Plant Biologists, recognizing his distinguished and sustained contributions to plant biology. Please join me in welcoming Dr. Erich Grotewold, and his presentation today is on regulatory activity of flavonoid pathway intermediates. With that, I'll stop sharing my screen. Erich, please go ahead and share your screen.
I'll do my best here. Here we go. Looking okay there? Okay. Well, Satya, thank you so much for that introduction and also for the invitation to talk to you all here. And I really want to thank Graf and Lars and many others in the team, not only for having started this series, but also for having sustaining it. I know that there's a lot of work in doing that, a lot of coordination and arm bending and so on. So very, very thankful because I think it's a great series. All right, so what I will do today is to tell you one story that I'm hoping many of you will draw some learning from regarding something that we did not realize really about flavonoids. Now, before I start, let me remind you a little bit. I know this seems far away now for those of us in the Northern Hemisphere, but the three major non-photosynthetic plant pigments are really the betalains, the carotenoids, and the anthocyanins. And these are really pretty much what provides all the colors to our lives in nature. Now, one issue that has been discussed quite a bit is what is the function of anthocyanins in fruits and flowers. That's quite clear. They provide attraction to pollinators and to seed dispersing agents. What is significantly less clear is what anthocyanins are doing in vegetative tissues. And this is something that, I guess as I'm getting older, it concerns me more and more because I realize that many of the models that are out there for explaining what anthocyanins do in vegetative tissues are less than satisfactory. And the three models that are really presented is that they're providing photoprotection or screen against light. But this has a problem, which is that anthocyanins absorb green and yellow light, which is really not so important for photosynthesis. Another function is that they serve as antioxidants, but the challenge with that is that they accumulate largely in the vacuole, where there is minimal ROS scavenging, and there is very little evidence about anthocyanins making it out of the vacuole in order to provide some sort of a antioxidant function in the cytoplasm, even less in the mitochondria, the chloroplasts, where much of the antioxidant function is really necessary. Another idea that has been put together is that it serves for buffering the carbon-nitrogen imbalance. And we do know that in many plant species, including Arabidopsis, and I show that here, and I will come back to that in a minute, if you grow seedlings, oh, sorry about that. If you grow seedlings in media lacking nitrogen and with significant high quantities of sucrose, say 3%, seedlings turn completely red. But the problem is that under most conditions, the amount of anthocyanins or the amount of carbon that's really sequestered into anthocyanins is quite minimal, less than 2%. And it's only when we think about anthocyanins accumulating to really massive levels over a very short period of time that one starts seeing some significant um, um, utilization of the fixed carbon into these pigments. So I don't think that this is really the main reason why anthocyanins have really accumulate in vegetative tissues. So once again, we understand much about what happens to reproductive organs. Kevin Gold, who is really the father of anthocyanins in many ways, called the anthocyanins in nature's Swiss Army knife, and proposed that they're really doing many different functions. Um, I would argue that perhaps we have missed one of the important functions that not necessarily anthocyanins, but anthocyanin pathway intermediates have. And that's what's going to be the topic of my talk today. And just to remind you, for those of you that don't think about flavonoids all that often, this is kind of the Arabidopsis flavonoid biosynthetic pathway. You can see that it starts from the phenylpropanoids, chalcone synthase, encoded by TT4 in Arabidopsis, the first committed step, which forms naringin chalcone, which is the substrate of chalcone isomerase. And then the pathway continues, naringenin being a key pathway intermediate that can go into different directions. Anthocyanins involve a number of other enzymatic steps, as you can see over here. And then Arabidopsis also accumulates tannins in the seed coat. Today, I'm going to be focusing mostly on this top part of the pathway. And what I want you to remember is that TT4 is chalcone synthase and that TT5 is chalcone isomerase. Now, for many years, we have been talking about flavonoids being secondary metabolites. And I guess that one of the characteristics of secondary metabolites is that there are no phenotypes most of the time associated with mutants in the pathway. And that's also true for most flavonoid mutants. And you can see here mutants in chalcone synthase, TT4, as well as chalcone isomerase, growing under what I would consider to be fairly normal conditions. However, just by chance, one of the members of my lab came across this other growth chamber in which suddenly anthocyanin mutants started showing some very, very dramatic effects. And you can see that while wild type accumulates in these conditions very high levels of anthocyanins, TT4 plants look fairly completely normal, whereas TT5 plants are completely stunted. And if you look in detail, you will see a lot of developmental defects associated with that. I didn't believe this initially, so Nan, who is the former postdoc that did most of the work, did all the due diligence to prove that in fact these mutations were in chalcone isomerase. And it took us a little bit of time to figure out what was really different between these two growth chambers. And it turns out that the growth chamber on the right is completely lacking far red light. And that the reason for that is essentially because they had forgotten to install the incandescent bulbs. So no far red light appears to be inducing quite dramatically the anthocyanin pathway, something that we didn't know. But most importantly for this is just by chance we discovered these developmental phenotypes associated with chalcone isomerase mutants. Now, it is known that the balance between red and far red light can induce anthocyanins, but we were a little bit concerned that no far red light whatsoever is not really a very physiological condition, despite the fact that it had this dramatic effect. So what Nan did then was to really try to phenocopy these phenotypes by using high light. What I called before as the growth chamber now 1 is about 80 micromoles per centimeter per second. And then when we switch to high light, you can see that you can phenocopy very well the results of not having far red light. Wild type accumulates significant amount of anthocyanins. TT4 looks normal, but then TT5 once again looks completely stunted. And none convinced me at that point that this was really TT5 because 3 complementation lines expressing TT5 from the TT5 promoter were able to fully complement the phenotype. And then when he crossed TT5 to TT4, what he found is that the phenotype was completely suppressed. And that provides some interesting evidence that perhaps it was the precursor for chalcone isomerase, the product of chalcone synthase, or a derivative that was responsible for these phenotypes. And that would be naringenin chalcone or naringenin chalcone derivative. And this is Nan, and I really have to give him credit for following up on something that I thought was a huge artifact. So it turns out that, in fact, these plants are not accumulating naringenin chalcone itself, but they are accumulating 4 major derivatives of of naringenin chalcone, a diglycoside, a malonyl glycoside, and then another glycoside. And this is all by using LCMS on those extracts, and I'll come back to that in a minute. So at that point, we were a little bit at a loss in trying to understand what was going on. We decided to seek help from RNA-seq, as we normally do. We, meaning the community, not just us. And what we did was to take the various mutants grown under high light or normal light, and then look at the genes that were differentially expressed. And one of the first things that we found quite surprising is the fact that the sets of genes that appeared to be affected by high light was quite unique to TT5, and you can see that over here. There's a very large number of genes being affected, which are unique to TT5, and many of those genes, actually some of the main categories of the genes that were being affected, including salicylic acid-mediated defense responses for the ones that were being induced, and then for many of the genes that were being repressed by highlight, corresponded to photosynthesis-related genes. That was very intriguing. So just as a quick summary, we see that the accumulation of naringenin chalcone significantly induces defense response genes, particularly mediated by salicylic acid, and then naringenin chalcone or a derivative, because we don't really see naringenin chalcone itself accumulating in the plants in the high light, only the derivatives, significantly inhibits photosynthesis-related genes, and this appears to be a process that's unique to TT5 because we don't see it in the TT4 mutants. And in fact, you can see that the levels of salicylic acid significantly increase, not just by light in TT5, but that those levels are significantly larger, what you normally see in the wild type. Interestingly, in the TT4 mutants, those are the chalcone synthase mutants, the levels of salicylic acid as well as the salicylic acid glucoside significantly decrease. So this was consistent with the idea that we were seeing salicylic acid responsive genes being induced. And there is quite a bit of literature that suggests that high levels of salicylic acid can actually cause developmental defects. So we were intrigued as to whether this might be the reason why we were seeing those stunted phenotypes in the TT5 plants grown in high light. So what Nan decided to do is a suppressor screen in which he tried to identify secondary mutations in a TT5 background that would suppress the developmental phenotypes when grown in high light. And I'm not going to take you through all the details through this, but we identified about 30 suppressors. I'm showing you some here, and the way in which Nan kind of triaged those suppressant lines was by doing LCMS of acid hydrolyzed extracts, which convert the naringenin chalcone into naringenin, and then seeing whether naringenin is being accumulated or not. And you can see that the vast majority of the mutants of the suppressor lines don't accumulate any levels of naringenin, suggesting that the pathway is blocked at some very early stage. But there were 5 suppressor lines were accumulating significant levels of naringenin or naringenin chalcone. 2 of those mutants that have a significantly reduced level of naringenin/naringenin chalcone compared to wild type ended corresponding to 2 new alleles of TT4. And this was validating our double mutant experiments that were showing the TT4, TT5 doubles were not having any developmental phenotypes. But what became really intriguing were the other three, because they corresponded to three novel alleles of UVR8. And you can see those here, completely different alleles of UVR8. And at that point, I was a little bit at a loss because, well, I knew that there was a UV receptor. I had no idea what UVR8 was like. However, luckily for me, Rob lives next door to my lab. So I went to Rob's office and asked, hey, Rob, tell me something about UVR8 since you and Dan cloned this gene several years back. I don't want to say how many years back. So then Rob told me everything he knew about UVR8. I'm just summarizing that knowledge kind of over here. Normally, UVR8 is in the cytoplasm as a dimer. It's considered to be the inactive form. When the plants see UV light, that dimer dissociates, the monomer moves into the nucleus and interacts with COP1 and with a number of other regulatory proteins and activates the entire UV response. The moment that UV light goes away, there is a group of proteins called the RUPs, which allow these monomers to form back to the dimer, and again, the pathway becoming inactive. And this in vivo, this is really fast. Within an hour or so of removing the UV light, all the monomer goes back to the dimer. So we were intrigued by why we were identifying UVR8 as part of the suppressor screen. And the first question we ask is whether these phenotypes that we were seeing were dependent on UV light. And the answer is no, because what you see on the top side here is the experiment done under the normal conditions, the highlight conditions, which does include about 50 microwatts of UV light. But then if you put a filter that completely blocks the UV light, you can see exactly the same phenotypes. So this was suggesting that whatever was going on, it's independent of UV, suggesting that perhaps what we were really facing was an UV-independent function of UVR8. The next thing we wanted to see what was happening with salicylic acid in the suppressor lines, given that this was kind of a lingering idea that maybe it was the high level of salicylic acid that was causing the developmental defects. But as you can see over here, the suppressor lines accumulates very similar levels of salicylic acid as the DT5 lines alone. And in fact, this is nicely induced by light. But this was really suggesting that the salicylic acid was not responsible for the phenotypes that we were seeing. The answer to what might be going on came from these experiments. And I will have to take you through this for a couple of minutes because they're a little bit complex. So as I mentioned before, UVR8 is normally a dimer. And when under normal conditions, there's a very, very little amount of monomer that can be found in the nucleus. Now, the community is very privileged to have an antibody that recognizes the interface between the two monomers. Essentially, it's hidden in the dimer, but it recognizes the monomers. So if you do a straight Western blot, non-denaturing, you can detect immediately the monomer very nicely, but you cannot detect the dimer. So in order to be able to detect the dimer, the trick that is used is to treat the gel before it gets transferred to nylon with UV light. And that exposes the epitope for the monomer to be able to recognize it. And that's what you're seeing in this slide. So in the first panel, you're seeing what happens in non-denaturing conditions in gels treated with UVB. So you can see that in 3-week-old plants grown under high light, essentially all that's there is the dimer indicated with a D. But as we keep on going with a high light, you can start seeing more and more of the monomer accumulating. You can run a denaturing gel and show that in fact the levels of UVR8 are completely constant. So this is not a variation in the levels of UVR8. So what these results were suggesting is that for some reason, the accumulation of naringean chalcone was somehow preventing the monomeric UVR8 to go back to the dimer, or alternatively, was dissociating the dimer into becoming a monomer. To distinguish between those possibilities, what we did was to use nanodifferential scanning for fluorometry, DSF, with recombinant protein, which we can purify very nicely. And then if you treat that with UV, it completely dissociates and provides the monomers. So what you see is that without UV and with UV, we're following the binding to naringenin chalcone in this slide and to a number of other flavonoids in the next slides to see which one is really binding with either the dimer or the monomer. And to make a long story short, but you can see that from the slide, is that naringenin chalcone interacts only with UVR8 following monomerization after UV treatment. If we take a mutant that is a constitutive monomer, we see very good interaction, even in the absence of UVB. But if we take a mutant that cannot form the monomer, essentially it's a constitutive dimer, irrespective of whether we have UVB light or not, it would not be able to interact with naringenin chalcone. So what this was suggesting to us is that the interaction actually happened with the monomer, probably not with the dimer, and that the binding to the monomer would be preventing it from forming back the dimers. The interaction is very specific to naringenin chalcone. We tested here a number of different flavonoids. I think I'm showing you here mostly camphorol, dihydrocampherol, leucocyanidin, dihydroquercetin, cyanidin, but we tested a few more. And the only one that seemed to be interacting with the monomeric form of UVR8 is naringenin chalcone itself. And then if we do, we use MST to be able to calculate an apparent dissociation constant. We find that the dissociation constant is about 12 micromolar, which is really a robust and strong interaction between a protein and a small molecule. And if we look with naringenin, which is kind of the immediate product of naringenin chalcone in the pathway, you see that this is about 100 times larger KD. So again, highlighting that this interaction is very specific. So in summary, what I've shown you that a higher level of monomeric UVR8, which presumed to be nuclear based on others' literature, but we have not formally proven, results from the accumulation of naringenin chalcone or probably derivatives. Naringenin chalcone binds very specifically and with a very strong affinity to the monomeric form of UVR8. And in TT5, it is probably the malonyl glucosyl naringenin chalcone, the compound that interacts most strongly with monomeric UVR8. And I'm not going to be showing you the results for that, but we think that this is the compound that's most active, not naringenin chalcone itself, even though, as you will see in the next slide, or in the previous slides, naringenin chalcone can bind to this, to UVR8 very strongly. But we have a problem, which is that if you grow Arabidopsis in the wild types in the greenhouse or in the growth chambers, there's absolutely no naringenin chalcone present, and neither are the derivatives accumulating. So which left at that point scratching our heads and thinking, what is the biological significance of all this? Okay, if we cannot detect intermediates because they get converted so fast into the products, why does this even matter? And for that, what we did was to ask the question, what happens if we grow Arabidopsis in perhaps more real conditions? And then we did this experiment in parallel in Michigan and in Hawaii. You can immediately imagine the advantages of doing this experiment in Hawaii. The Michigan experiment didn't look that clean. You can see what are the conditions of the plants of their growth in these conditions. And what I did is, so this is a total flax, this is the temperature, and this is the amount of UVB. And what I put here with a straight line in each one of the graphs is what were the conditions that we used in our growth chambers. You can see that the amount, the flux of light, total light, as well as the flux of UV is dramatically larger in outside than it is in the growth chamber. And on the left, what you see is the phenotypes of all these plants grown either in the presence of a UV filter or in the absence of UV filter. And what you can see is that, as we have seen before, TD5 has this dramatic phenotype. Now, what happens with the accumulation of the naringin chalcone and derivatives, well, now you can start seeing the accumulation in the wild-type plants. The levels are not incredibly high, but they're way higher, several orders of magnitude higher than what we normally see in the growth chamber. What's intriguing, though, is the fact that UV, contrary to what the dogma is, doesn't seem to be having much of an effect on the accumulation of this pathway intermediate. So it made no difference if we had a UV filter or not, a very minor difference. What happens with the proteins? You can see now that, again, experiments done exactly as before. The only thing I'm not showing here is the dimers. We're focusing on the monomers. You can see that there is a significant amount of monomer present in the mutants as well as in the wild types. Again, there might be slight difference in the amount when UV light is present compared to when UV light is not present. But the reality is that in these conditions, we can detect significant amounts of monomeric form of UVR8 accumulating. Interestingly enough, if you look at TT4, you can see, so remember this one, this is a chalcone synthase mutant. There is no naringin chalcone accumulating of any sort. You can see that actually the amount of monomer that forms is significantly lower than what you see in the wild type. And this is even more dramatic when we have a UV filter. So at that point, we were trying to understand what was the mechanism by which this was happening. And one idea that crossed our mind is that perhaps what was happening is that by keeping naringin chalcone stabilizing the monomeric form of UVR8, we were creating this constitutive UV response, which is known to have a negative impact on the growth of the plant. So to see if that was the case, what we did was to compare the genes that were affected by UVR8 with the genes that were affected by TT5 under highlight. And what you can see over here is that the overlap is absolutely nothing, which means that UVR8 or UVB responsive genes and those induced by TT5 are really not overlapping at all. So which means that what's happening here is not really just a constitutive UVB response. So what we did next was to do RNA-seq on plants comparing Columbia and UVR8 mutants under highlight, and then TT5 under highlight and normal light. And this became really interesting because what we found is that now we could see a lot of genes that were, and I should also mention, this is absent in the absence of UVB. So what we are really revealing here is an effect of UVR8 that has nothing to do with UV response. And you can see that the mutant UVR8 has very few genes. This is what, about 70 genes that are being, a little bit more, 100 genes that are being affected, okay? But now we found a very significant overlap with the genes that were affected by TT5. And in fact, what we found is that from the 27 genes upregulated in TT5, most of those genes were also upregulated in the UVR8, okay? And the vast majority of those genes appear to be involved in disease-resistant functions. So again, this was unexpected, but what we think is that under normal conditions, low light, doesn't matter if there's UV light or not, UVR8 shuttles a little bit between the cytoplasm and the nucleus, keeps a very low levels of UVR8 in the nucleus. When UV comes on board, then there is a significant movement of UVR8 into the nucleus. It does a lot of things, but one of the things that happens as well is interaction with naringin chalcone. And this interaction with naringin chalcone affects, in addition to all the other genes which are TT5 independent, affects also a set of UV independent UVR8 genes that are involved with disease resistance. And we think that these are the genes, this set of genes over here, are the ones that are responsible for the phenotypes that we see in the mutant grown under highlight conditions. So this is our work model right now. It has a lot of testable hypotheses that we are following right as we speak. So in summary, what I've told you is that Vegetative anthocyanins serve as indicators of abiotic and metabolic stress, but really we don't understand what is the function in vegetative tissues. We have wanted to use Arabidopsis flavonoid mutants as a way to understand better what is the function of anthocyanins, but clearly the mutants are doing things on their own. Intermediates in the pathway have functions that we did not anticipate. One of those functions is shown over here, which is this ability of naringin chalcone derivatives to be able to interact and stabilize monomerically active UVR8 in the nucleus. We also find out something that we don't know what it means, but that accumulation of naringin chalcone has a very dramatic effect on salicylic acid accumulation, but this is completely independent of UVR8. Our studies also suggest that UVR8 has a number of UV-independent functions, and that some of those functions, at least, appear to be modulated by the accumulation of flavonoid pathway intermediates. And I want to remind you that this is just one example of other flavonoid genes that we have found before, that when the mutations that it create have effects that seem to be suggesting that many flavonoid pathway intermediates play regulatory functions at different levels inside the cell. So maybe going back to the idea about the Swiss Army knife, it's not really the anthocyanins, the ones that are serving this function, but it's flavonoids in general. I want to finish thanking, of course, Nan. It was his perseverance in this that made this happen. Sunny, who was a postdoc in the lab at that point as well, and she's now faculty at Seoul National University, for helping out with a number of the protein small molecule interaction assays. Tatiana for the bioinformatics, and then Emily also for helping out with protein purifications and analysis. And of course, Rob for a lot of advice and suggestions on this project. Tony at the facility for helping us out, figure out which compounds were really important. And then Ola for sharing with us the NanoDSF and the MST. And of course, the funding agencies. And thank you very much for your attention. And I'm going to stop sharing so I can see you all.
Thank you for such a wonderful presentation. The floor is now open for questions. Anybody have questions? Gaurav, yeah.
Thanks, Erich, for a nice talk. I was wondering why you think naringin chalcone interacts with UBR8 in the nucleus rather than as with the dimer in the cytosol.
Yeah, we don't know if the interaction happens in the nucleus or in the cytoplasm. What is known is that immediately when UVR8 dissociates, the monomer moves to the nucleus. So there is very little, from the literature, there is very little monomeric UVR8 in the cytoplasm. So it is based on that we think that the interaction with the monomer is happening in the nucleus, but we don't have the evidence.
Okay.
A quick follow-up question on this interaction. Do you think that this is like a broad, like mechanism is broad, conserved across species, and also mainly environmental conditions? Is it like, I'm thinking, is it like in different environmental conditions, do you think this mechanism is conserved?
Yeah, those are great questions. Regarding the first one, I was hoping that we would have the data to show you. We're doing now the same experiments with a number of monocot UVR8s to see if they can interact with naringin chalcone. But unfortunately, they have been shown to be a little bit more problematic for purification than what we anticipated. So I don't have the answer for you yet. Regarding the environment, absolutely. One experiment we really need to do is to see how temperature is affecting this process.
Right.
Because it is known that UVR8 is sensitive to temperature. And anthocyanins and flavonoids in general are also sensitive to temperature. So yeah, so that's on my bucket list.
Thank you. Gaurav has one more question.
Yeah, if I may. So in terms of, so flavonoids frequently come up as the top binders for human proteins in computational studies, for example. Um, and they, there is this therapeutic potential that is ascribed to flavonoids. That's not just the antioxidant functions. So at the levels that they accumulate here under UV stress or found in plants, uh, are you able to say anything about the therapeutic potential or the concentrations, whether they are relevant from a therapeutic perspective?
No, and as I showed you, the levels of naringenin chalcone under normal conditions seem to be fairly low in the plant. Now, of course, we have no idea where in the cell we are looking at those levels, right? You could easily imagine that there are specific domains within the cell where the concentration is much higher, and those might be the ones relevant for the interaction with UVR8. But I have no idea. Regarding your first part of the point, it's true that many flavonoids appear to be bioactive in animal studies. Initially, many flavonoids were supposed to be interacting with proteins non-specifically, just because that's a presumed characteristic of polyphenolic compounds. We have done some studies looking at the binding of flavonoids in humans, and what we found is that in reality, the interactions seem to be very, very specific. Even kinases, which have been proposed to be a major target of flavonoids, don't seem to be the main target at all when you're working at a reasonable concentration of flavonoids.
Thank you.
We have a question in chat from Maddie Larson. Great talk. Do you expect that small molecule interactions like naringenin and chalcone derivatives might also influence dimerization and downstream signaling of other photoreceptor types?
I have no idea, Maddie. I did not expect this. So if I were to make guesses, 2 years ago, I would have never guessed this either. So I really don't know. It would be interesting to test. We have not any evidence from what we have looked here that any other photoreceptor appears to be affected. Okay, but in reality, we didn't even look for that. Now, regarding the ability to affect the dimerization of other proteins, yes, we do have evidence that some flavonoids can affect the way other proteins dimerize. But this is mostly related to domains present in transcription factors.
Any other questions for Erich today? All right. If not, thank you so much for your wonderful presentation and interesting paper. Thank you so much, Erich.
Well, thank you, everybody. Thank you, Satya and Graf.